This unit that we're starting is on reproduction, and we want to take a second and really think about what is reproduction. A really simple definition of reproduction would mean to make more. And so then the question is, what does this more look like? Is this more identical, or does this more have variety? And so that'll be one of the key things that we'll talk about in this unit, is the difference between asexual reproduction and sexual reproduction. So when we look at making more, we want to stop and think, why is an organism, why is this happening in biology? And so when we make more, we might be making more organisms. So an important reason for reproduction is to continue the species. An individual has a finite lifetime. It won't live forever. And so sometimes we are going to be continuing the species. Another reason to make more might be to grow the organism, or perhaps even to repair part of that organism. When we look at asexual reproduction, we're mainly going to see that asexual reproduction is going to have to do with either growth or repair. But some organisms are going to use asexual reproduction to continue the species. So we do want to take a second to talk about what it looks like when we use asexual reproduction to continue the species. When we use asexual reproduction, a characteristic of asexual reproduction is that the quote-unquote offspring are identical to the parent. Now, this will apply whether we're talking about making more organisms or whether we're talking about making more cells. So we can use asexual reproduction to make more organisms or to make more cells. If we're making cells, we're usually trying to grow the organism or repair the organism. So when an organism, a living thing, uses asexual reproduction, the offspring, whether those offspring are cells or new organisms, are going to be identical to the parent. And what we mean identical is we mean genetically identical. The genome, the DNA sequence is going to be identical. So all that we're really doing is copying that organism. And sometimes we actually call this cloning. Now, we'll talk a little bit more later when we get to DNA technology about the actual process of cloning, like creating the cloned sheep dolly. For right now, we just kind of want to associate the word clone with identical and with asexual reproduction. We're going to see a couple different varieties of asexual reproduction, so that's where we're starting off with here. One type is in vegetative propagation, and so these three pictures are all examples of vegetative propagation. Vegetative propagation. And what we'll see is that somehow the organism is going to create a new clone of itself, and that's how it's going to grow and regrow. One of the advantages of asexual reproduction is that this can occur very quickly. So whenever the environmental conditions are right, the organism can very quickly clone itself and create offspring. There's no need for a mate. And so you can see the advantages of asexual reproduction would kind of contribute to evolution, that if you can create lots and lots and lots of yourself under ideal situations, then that organism is going to be very successful at surviving, which is the main tenet or purpose of evolution, is being able to survive. Another thing that we're going to see is this concept of cell division. So we'll talk about binary fission. This is really going to be an example of cell division. But we do also want to talk about something called budding. And budding is where you create a mini-me. It's where you create a smaller version of the organism, and then that will break off and become a larger organism. It will grow all on its own. A hydra is actually an example very similar to like a sea anemone, so it is actually a marine animal that is doing this. It's a jellyfish, a form of Cnidaria, a type of jellyfish that's actually anchored to the marine floor. So what's important about this is that we see a variety of uses. So asexual reproduction is used by all living organisms in some way or another. And so sometimes it's used for different purposes, sometimes it's used in different manners, but we do see that all living organisms at some point are going to use asexual reproduction. When we talk a little bit about the repair, what I'm going to ask you to do is click on this picture. It'll take you to a really short video about regeneration, which is one of our other examples of asexual reproduction. And so what we're going to see in this video is starfish regeneration. And I would like for you to list some things that stand out to you. So you might need to pause the video, you might need to watch it twice. It's only about a minute. But there's some really interesting things that should kind of make you think. So if you want to pause this video and click here on this link that's hyperlinked to this picture, then you can go through and watch a little bit about regeneration. We want to move on. So after you have watched the video about how the starfish regenerates, we want to look at how does a starfish actually regenerate? It's not magic, right? The arm doesn't just grow out of anywhere. The arm actually regenerates because the cells are going to copy themselves. So cell division, interestingly enough, in science, division and multiplication are the same thing. So in cell division, we're making new cells. And asexual cell division is going to make identical copies. And so why would we use this? This is going to be used mainly when we're talking about the growth of a multicellular organism. So let's stop there for a second. Growth of a multicellular organism. Organisms aren't just one big cell. They are many cells. So this concept of multicellular, the reason that we need to be made of many cells is that the smaller units function better or more efficiently. So we know that from the size and volume. So we want the surface area to be large and the volume to be small. We saw that when we talked about the cell membrane. We need a large cell membrane so things can enter and exit the cell. We don't want a huge volume. And so what we see is that asexual cell division is mainly going to be used when we're talking about the growth of a multicellular organism. We want to keep the DNA the same, but we just want to make many and many more copies. One of the interesting things that will happen in multicellular organisms, though, is the concept of differentiation. And we'll talk much more about differentiation in the coming units, but the main concept is that when you have many cells, they can perform specialized functions. And so what you see here is a picture of a stem cell, a cell that can divide, and then as that cell matures, that cell will take on a very specific role. So what I would like for you to do at this point is take a second and do a little Googling. Find an example in the human body of cell regeneration. If you Google human cell regeneration, you'll find a lot of great resources. Just be careful of what source you're choosing to use. So we do want to stop and think a second about what does this look like in a multicellular organism. Yes, I'm making all these exact copies, these daughters, these identical daughter cells. But you'll want to pause the video here as well and really look at what will actually happen to those cells. So after you've done your Googling research, we do want to talk a little bit about how does this process of cell division happen? How is it that dividing actually equals multiplying? That doesn't make much sense. How does dividing one cell into two actually make sense? Well, the concept is that what happens is that we actually begin by replicating or copying the DNA. We know that DNA is really what codes to make a cell a cell. So if we're making more cells, we have to make more DNA. And so this section here is really all about what is going to happen to that DNA. We'll cover DNA replication and how it happens a little bit later, but we do want to know that the reason we can divide a cell is because we're actually multiplying it or copying it. So while cell division actually divides the cell into two, we're not losing any information. So no loss of DNA. We're going to maintain the same information. One of the really important things to notice in a cell is what that actually looks like. So we're very familiar with the DNA double helix, but what we need to know is that DNA is actually packaged into a chromosome. So the double helix is actually coiled around some proteins to form a structure called a chromosome. And we'll talk more about the packaging of DNA in the next unit as well. But we're going to see these pictures of chromosomes, and we need to know what they mean. Right before cell division, this chromosome is going to be copied and produce a doubled chromosome. And that is the picture that you actually see here. And so if we could turn this chromosome, you would see something like this, almost butterfly structure. And we want to take a second and get some of the vocabulary down here. What's actually happening in this picture is that you have what used to be called a chromosome, right? So here is a single chromosome. After I have copied it, I have a doubled chromosome. So I call the whole structure a doubled chromosome. And what this means is that the left side and the right side are identical copies of the DNA. And we call these sides sister chromatids. So we use the word chromatid when we're talking about one or the other side of a doubled chromosome. So single chromosome, doubled chromosome, and now I call the two sides sister chromatids. And then we have this darkened region here, and this is called the centromere. And this is going to be a region where there are proteins attaching the DNA copies together. And we are going to see that this centromere region is going to become very important for what we do to these copies, when we will actually pull these copies apart. So, coiling the DNA up into a chromosome, and as it's coiling, we're actually replicating it so that we end up with a doubled chromosome. The significance of this chromosome packaging is going to be that the chromosome is now condensed, and it is easy to move and then divide the copies. So what we need to be really good at is looking at the difference between singled chromosomes and doubled chromosomes. And we're going to mainly be focusing on the example of humans. And so we'll look at how there are actually 46 chromosomes in any human cell. But when we prepare for cell division, we actually